Alright, hi, I'm Luke, and I'm excited to tell you about my project, which is the variation in the abundance and impact of nest ectoparasites between two passerine species, and those species that were studied were tree swallows and house wrens. So first, it's important to understand what can influence the health of a species, and there are numerous factors in an environment that may um, influence or change the health of a species or of an individual in a species. So you have things like food availability, habitat, how often they're getting predated, and there are other examples. Often these are shared across numerous species, so different species can have the same preference for habitat, and they will fight each other for access to that habitat, or they might like the same types of food, and they'll try to fight each other for food. And then one predator may impact multiple species, sort of um, their food sources. However, not all species are impacted in the same way, so there are some adaptations that some species have that can mitigate these impacts of factors on their health. For example, if you look at the factor of water availability, you have certain animals or species that can become resistant to more drought conditions and may not need water as often as other species who rely on water more. And then if we're thinking in terms of the factor that I was studying, which is parasitism, more specifically ectoparasitism, and I have a slide briefing on the different forms of parasitism, but for example, the tree swallow, they have been shown to have some sort of immune response to certain ectoparasites, and how that works is they have an antibody response that when they become bit, their skin becomes inflamed so that any ectoparasites that are on the surface of their body and trying to bite through that skin to access their blood, that skin sort of stretches out like a balloon and it's harder to bite through and access what they want to get, which is that blood. So now that we sort of understand the host species, let's get into the sort of the parasitic side. So there are three different ways that a parasite can function on their host. So first, we'll have ectoparasites, and that's the one that I studied a lot and what the focus of the project was on, um, which are parasites that live sort of on the surface of their host on their body, and then they usually bite through the skin to access the blood or other ways. And then you have endoparasites, which are similar, but they will enter the body, enter the host's body either through ingestion or some other mean, and then they'll get their nutrients. That way, they can dig their way through the walls of their host in like the intestines and get their nutrients that way. And then the third form, which is sort of unique to avian species, is this thing known as brood parasitism. So basically, instead of a bird species putting all that time and energy into making a nest, they will instead go to other nests already made by different birds, and they'll just lay their eggs um, as a way to, spread their DNA, spread um, their future offspring, but without needing to impose the cost of building a nest. And these can kind of get a bad rap because their nestlings can um, sort of be stronger and can kill or harm the nestlings of the host. So now that we understand what species there are, what strategies there are, the species I was so now that we understand some of the mechanisms that parasites have for getting their nutrients, I want to focus on the parasite I looked at for my study, which is Protocalifora coelea, and this is a species of blowfly. Typically, a lot of the blowflies are like normal flies, or they'll look for like dead things, but there is a genus which are most commonly known for their ectoparasitism of nestlings, and that is the Protocalifora genus. So these are common nest ectoparasite. You can find them in a lot of different species nests. However, most of the studies agree that location of the nest, so sort of their habitat and environment, is what determines if a nest will be infected and what species of Protocalifora will be affected by. And there have been some studies I've seen where species may play a role, but overall the common agreement is that it's location and not species. And as I've discussed before, some host species have defenses, sort of tree swallows and their immune response that sort of inflate their skin and prevent the biting of the ectoparasite larva. So now let's get into the methods. We had a lot of nest boxes set up at several sites on the Duluth, Minnesota area, and these photos show you sort of what they looked like. We had a marker on each side of the box so we could figure out what side it was at and what specific box. And then you see this sort of metal cylindrical thing around the base, and that would go around the pole to prevent any squirrels or raccoons or things from climbing up and predating on the nestlings. So overall, I was able to collect 27 house wren nests and 46 tree swallow nests that were able to be dissected. So the process of dissecting these nests could be quite intensive. So I would take them apart and sort of separate their materials into different piles. So I had a hard material pile, which would be mainly of sticks, and then I would have a soft material pile, which would be pine, grass, sort of this softer building material. And usually the tree swallow nest could be knocked out in about 15-20 minutes just because you have to be careful. But the house wren nests are so big and there's so many sticks to separate that one nest could easily take up to an hour and even more, just depending on how complex it was. And then for each nest, a subset of nestlings that were able to hatch and get to the day 11 stage were dissected so we could take body measurements, including stuff about their organs that we can sort of link to their health and have that as a stand-in for their health. And nests were classified as parasitized or non-parasitized depending on if Protocalifora larvae were present at the time of dissection. Alright, before we get into the results, I want to show you my hypotheses so you can kind of be on the same page of what I was thinking going into this project. So I hypothesized that tree swallows will have a lower parasite density compared to house wrens, and that parasitized tree swallow nestlings will show less variation in body measurements compared to parasitized house wren nestlings. And this mainly stems from, that adaptive response that was established in tree swallows to ectoparasitism and other studies that showed house wren nestlings sort of lose more blood when parasitized compared to tree swallows. So if they're losing more blood because that's what these ectoparasites are feeding on, we would expect them to sort of have a lower expectancy of growth and would show more variation in these body measurements. Alright, and now let's look at these results, and they were not what I was expecting. So this top graph here shows the species on the x-axis and the number of Protocalifora found in the dissected nest on the y-axis. So this results from summing all the Protocalifora larvae we found in dissected nests for that species, and there's a pretty big difference. So tree swallows ended up near the 440 mark, and house wrens just had one infested nest with two larvae found in it. And this bottom one shows the probability of a nest being infected by Protocalifora. So we have that probability on the y-axis and the species on the x-axis. And as you can see, a very big difference again. The bars represent plus or minus 1 standard error. And these results were really unexpected to me because I came in with this idea that it's location that's impacting these infestations and not species. And these next boxes were in pretty much identical habitats, and even at one site, it would be in a row. We'd have 2 tree swallow nests and 1 house wren nest, and it seems that the Protocalifora would sort of skip over that house wren nest and go to the 2 close by tree swallow nests, which was kind of interesting. And this just kind of goes against the main idea, which has been sort of looked at in other studies, that species may play a role in sort of Protocalifora infestation. And so sort of what I found is it seems species, not location, plays a bigger role in infestation, or if not a bigger role, at least some role that can't be just not accounted for when looking at the possibility of being infested. My results match other studies I've seen, but not all. It seems there's kind of I wouldn't say divide, but there is sort of an idea that location plays a role and species may or may not play a role. And I just think more research is needed in this field. In order to be effective in terms of conservation, we really need to understand what sort of pressures a species is going through. And it could be possible that these Protocalyphora infestations happen differently across different populations, and it just calls for more research of each population that we can manage to observe. And this photo sort of depicts what a Protocalyphora larvae looks like. And an interesting behavior, or I don't know if it's a behavior, but it's something my lab partner and I encountered, is we would often find the Protocalyphora larvae sort of cocooned in that nest material or like on feathers that we find in the nest. And it sort of is like a caddisfly when it creates a casing, but I wasn't able to find any mentions of this. So I'd be really interested in looking into this and seeing uh, why this is occurring or if has this been seen before. It probably has, and it may result from my methods of we would freeze the nests before dissecting them just so all the Protocalyphora larva is killed and there isn't that risk of like metamorphosizing and opening the bag and losing the flies. So it is possible that this is a response to those cold temperatures and they'll sort of try and cocoon themselves. Um, I have read that the adult flies can overwinter in a nest, but not the larva. So I'm not sure if it's resulting from the cold temperatures or if this is something they do when they're ready to metamorphosize into an adult fly and they just sort of cover themselves to prevent any predation or notice. And so I'd like to wrap up my presentation by giving thanks to the Great Lakes Toxicology and Ecology Division Laboratory for hiring me as a field crew member for their avian team, and the project lead with Matthew Ederson. And these are the team members I worked with. And this was a really amazing experience. It's opened my eye to ways of research and field experience that I was not thinking about before. I've worked as a field crew member for 2 summers, and so my first summer I was just observing and helping, but then I noticed these larvae in the nest, and it got me really interested, and that's sort of what led to this Europe project. I was able to do it while working for my second summer, and I just have to give extra thanks because I didn't have to set up any of the nest boxes. I was able to help them with their project, and then they weren't going to use the nest, so I was able to take them and observe sort of these patterns. So yeah, that's it.